Hi, everybody. Welcome back. So we will restart the sessions. It's my pleasure today to introduce Sandrine Dudoit, who is professor and chair of the Department of Statistics and professor in the Division of Biostatistics and at the School of Public Health at UC Berkeley. Her research interests regard high-dimensional inference and include exploratory data analysis, visualization, loss-based estimation with cross-validation, and multiple hypothesis testing. Much of our methodological work is motivated by statistical inference questions arising in biological research, and in particular, the design and analysis of high-throughput microarray and sequencing gene expression experiments. More generally, I would say I'm very pleased to introduce some statistical expertise at BayLearn. My experience in my department at AT&T is that I really benefit from being exposed to ideas from statisticians as much as I am from ideas from machine learning. So I think it's great to also have this point of view in our conference. So our talk today will be about learning from data in single-cell transcriptomics, where she will discuss statistical methods and software for the analysis of single-cell transcriptome sequencing data to investigate the differentiation of olfactory stem cells.
Good morning. I would first like to thank the organizers for inviting me to participate in this symposium. Given that we're all in the Bay Area, I hope we'll get a chance to meet in person sometime soon. I will talk about learning from data in single-cell transcriptomics. I've had a great time working on this topic with my PhD students and postdocs, Philippe Boileau, Nima Hejazi, Fanny Perraudot, David Hérisseau, Hector Roux de Bézieux, Kelly Street, and Koen Vandenberg. Special thanks to John Nye, my collaborator of 20 years, for motivating our work in this area. And I also like to acknowledge my collaborators in statistics and CS, in particular, Lieven Clément, Elizabeth Purdom, Jean-Philippe Vert, and Nir Yosef. Microarray and high-throughput sequencing assays have revolutionized biology by allowing the study of biological processes on a genomic scale. For example, with single-cell transcriptome sequencing, or in short, RNA-seq, we can measure the expression levels of an entire genome at the resolution of a single cell. And this resolution is really critical for many biologically relevant questions. For example, for the study of stem cell differentiation, or for the discovery of cellular subtypes in the brain. RNA-seq studies provide a great example of the range of questions we encounter in data science, where the data are complex in several ways. They're complex in terms of their dimensionality, where we face both the small n large p setting as well as the large n large p setting. There's various levels of processing, from raw images all the way to process matrices of read counts. We have various types of data: images, character strings, integer matrices, graphs. We have metadata, or data on data. In this case, this would be data related to the genes and data related to the cells. The data are sparse. There are also large technical biases due to batch effects, but also to sequence composition effect. And there's often low signal to noise. So before getting to the specifics of the biological context, I'll summarize the main steps in the data science workflow, because this is basically the process that we followed in our collaboration with the Nye Lab. So it all begins with a driving domain question. In our case, this would be the study of stem cell differentiation in the brain. The next step is to frame and translate a possibly vague domain question into a data-enabled statistical question. And this step is essential and far from trivial, yet it's often overlooked. In terms of pure statistical inference, the question is the parameter that we try to estimate or test hypotheses about. A very important aspect of the workflow is study design. It's important to design the study properly to make sure that we can actually answer the question that's motivating the study. This relates to notions of bias and confounding. And also we want to answer the question as accurately as possible given available resources. And this relates to sample size and variance issues. Then comes data collection, storage, and accessing the data. A very important step which we spend a lot of time on is exploratory data analysis for quality assessment and quality control, also to identify the main features of the data and whether there are problems with these data. So it's really critical to look at data, otherwise we fall into problems of garbage in, garbage out. And it's also important to use the right types of graphical summaries. In other words, go beyond histograms and pie charts that are still quite commonly used. Then comes data pre-processing. In our case, we spent also quite some time on that. I'll mention some of the methods we've developed for data normalization. And this seemingly mundane step can have a very large impact on the results of downstream analyses. Statistical inference and machine learning, that's the typical entry point of statisticians and computer scientists. That's our comfort zone, where the data have this familiar format of a matrix X by X and by P. This is a very important step, very challenging, but we have to realize it's only one aspect of an entire data science workflow. And to do this step properly, it's very important to be aware of the context and the steps that came before it. In particular, we need to think about the interpretation of the probability statements. For instance, what are the sources of randomness? What are the sampling units? Also, we need to ask ourselves, does the method actually answer the question that initiated the study? In other words, we want to be aware of hammers looking for a nail. This is very common to see that in the field of computational biology, and also of unrealistic modeling assumptions. Next come the interpretation, the validation, and the translation of the results into domain insight. And this often leads to entirely new domain questions. So this process, the data science workflow, is highly iterative. There's a constant back and forth between the different steps. For instance, we keep refining the question, refining the study design, and the validation. It's a very collaborative and interdisciplinary process. Feedback from the domain experts is essential throughout. Of course, we need software for computing with the data. Computational reproducibility should be a sine qua non to these studies. By that I mean we should be able to reproduce the computational results of a study given all the ingredients, the data, the text, the software, and the code. We need to do this just because it's good practice as part of the scientific method. And this still tends to be confused with a much more ambitious notion of biological reproducibility. And I really like this quote from John Tukey. I think it's just as relevant now as it was 50 years ago. Far better an approximate answer to the right question, which is often vague, than an exact answer to the wrong question, which can always be made precise. So a few words about the biological question that motivated all our work. So our collaborators in the Nye Lab are interested in elucidating the cellular and molecular mechanisms that underlie stem cell mediated development and regeneration in the olfactory epithelium. This has applications to the development of treatments and therapeutics to ameliorate neural tissue damage and degeneration. So they're focusing on a particular type of adult tissue stem cell, the horizontal basal cell, or in short HBC, and the p63 protein, which acts as a molecular switch. When p63 is abundant, the HBCs keep regenerating and self-renewing. And when p63 is knocked down, when it's absent, then the HBCs begin differentiating. So in order to investigate how these horizontal basal cells differentiate, Donnell performed single-cell transcriptome sequencing for control mice that had regular levels of P63 and mice for which the P63 gene was knocked down. So in a nutshell, here's how single-cell transcriptome sequencing works. You isolate individual cells from an organism, then you extract the RNA from these cells, you chop up the RNA into small pieces, and then you sequence these fragments to identify the sequences of bases, the A, C, G, U's. And once you have these small fragments, you map them back to the genes, the transcriptome. And the main idea in these experiments is that the number of reads that fall into a particular gene region represents the expression level of that gene. So from now on, the data that we'll be working with are very large matrices with rows corresponding to individual cells, hundreds or thousands of cells, and columns corresponding to genes. And the entries are these read counts or expression measures. We should be aware that before getting to that point, to this entry point, there's a whole bunch of pre-processing steps like image processing, base calling, and read mapping. But I won't focus on these steps right now. I'll take these for granted and begin working with these matrices. There's still a lot of EDA and pre-processing to be done, as you'll see next. So the question that we're trying to address here are, what groups of cells are present? How do these cells differentiate? And what are the genes responsible for the differentiation? So here's a look at the organism that we're looking at. This is a mouse's head. The olfactory epithelium is right here. And here is a diagram of the different types of cells that you have in the OE. The HBCs are in green. Under normal conditions, they self-renew. And then when P63 is knocked down, they start differentiating in various types of cells, including olfactory neurons and sustentacular cells. So this bar plot summarizes the experimental design. The height of the bar represents the number of cells. Each bar corresponds to a technical batch, and the bars are colored according to the biological conditions. So the biological condition would be whether it's a control mouse or a mouse that had P63 knocked down, and we look at different time points. Okay, so something that should jump right away at you is that we have a nesting problem here. The batch effects are nested within the biological effects, and we'll have to deal with that later on in the analysis of the data. So rather than focus on one particular question in depth, I'll give you a survey of the methods that we've developed to address the range of questions along the RNA-seq data analysis workflow. I'd be happy to provide my slides, and also links to all my papers are at the end of the slides. So of course, EDA, we want to look at data. Here in gray, I've indicated the software packages that we've developed for each of these purposes. Normalization, we've developed methods for normalization as well as performance assessment and selection of appropriate normalization procedures. Dimensional reduction is very commonly done in these RNA-seq experiments for EDA, but also for looking at the result and also as a preliminary step to clustering. So we've developed a method for sparse contrastive principal component analysis to remove unwanted variation and also extract sparse, stable, and interpretable signal. Quantifying expression levels. So bringing together a whole bunch of observations that we've had in EDA and normalization, we've developed a general framework for expression quantitation based on a zero-inflated negative binomial model. The main idea here is that we use the parameterization of the negative binomial mean and the zero inflation probability to accommodate experimental design features, so nesting in the case of the OE experiment, to also perform normalization by adjusting for known and unknown factors of unwanted variation, And also through this model, it's some sort of factor analysis, we can perform dimensional reduction. And this model yields weights also that we can use in standard bulk RNA-seq methods. Bulk RNA-seq is where you look at RNA for aggregates of cells as opposed to RNA for single cells. So I won't spend much time at all here on the sparse contrastive principal component analysis or the zero inflated negative binomial model. Instead, I'll focus on the rest of the talk on normalization, cluster analysis, and then the inference of cell lineages. But I just want you to be aware that we have all of these methods available. So let's take a look at data. So here's a bar plot of the proportion of genes with zero count. And the bar plots are colored according to batch. So there's a very serious zero inflation problem. We have up to 80% of the genes that have zero counts in a given cell. This is really extreme sparsity. So observation 1. Here are box plots of QC measures for each of the cells, and I've stratified the box plots by batch. So these QC measures reflect properties of the sequence alignment and of the sequence composition of the reads. The main message here is that quality varies between batch across all of these different QC measures. And this next figure here, this is a scatter plot of the first principal component of the QC measures against the first principal component of the read counts. Those are our expression measures. So the take-home message from these plots is that these quality control measures are associated with the read counts. Those are the expression measures of interest to us. Okay, so this is something we'll have to account for also when we try to normalize the data later on. And then the last plot, among many others that we typically look at, but I don't have time to show right now, is looking at the gene level read count. So the data structure we have now, again, is this matrix with rows corresponding to cells and columns to genes. And here, the plot on the left are box plots of the log counts. One box plot for each cell. We have about 500 or 600 cells. And the box plots are colored by the batch. So the batch represents technical variation only, not biological variation. So again, the zero inflation is flagrant here, but you can see that the expression levels vary greatly, not only between batch, but within batch. And within batch, we should only be looking at technical variation. So you want to really stabilize these measures within batch so that they're comparable. So this brings me to normalization. The goal of normalization is to adjust read counts for gene level and sample level unwanted technical effects in order to allow meaningful comparisons of the expression measures both between genes but also between cells. Normalization is essential before any clustering or differential expression analysis to make sure that when we see differences in the expression measures, these are truly due to biological effects of interest and not technical artifacts. And normalization is even more important for single-cell RNA-seq than bulk RNA-seq data due to the increased technical noise from single-cell experiments and also the zero inflation. So before we embark on all these data transformations and normalization, the question is, does it matter? Does normalization matter? Well, it does. The choice of normalization can actually have a greater impact on the results than the choice of downstream machine learning method for inferring differential expression or for clustering the genes or samples. So if it matters, then what method should we use? Well, that's the tough question. It's really not obvious. The choice of an appropriate normalization is highly dataset dependent. So what we need is a data-driven approach and control samples and genes to select a proper normalization procedure. And this is the question that we're addressing with the scone framework. So this is a framework for normalization of RNA-seq data, where we implement a range of normalization procedures, ranging from very simple scaling procedure like D-seq, TMM, or upper quartile normalization to regression-based methods on both known and unknown factors of unwanted variation. And then we've also developed performance metrics for assessing and comparing different normalization procedures. So these metrics are based on the clustering of samples according to factors of wanted and unwanted variation. They're based on the association of expression measures with factors of wanted and unwanted variation, and also between sample distributions of expression measures. And this framework, we look also at various numerical and graphical summaries of the reads counts. There's a Shiny app for examining for the results at various stages of normalization. This is just a quick screenshot of the Shiny app. I'll just show you briefly the results of applying scone. So testing about 70 different normalization procedures and selecting them based on these performance metrics. So this is the after plot corresponding to this before plot, the box plots of the log counts. So normalization does what it's supposed to do. It centers the log counts. And also it reduced the association of the normalized counts with the QC measures. Okay, cluster analysis. Clustering of genes or samples is a very common task in omics data analysis for the purpose of discovering new classes of genes and cells, but it's also very useful for EDA, for quality control, as well as an input to other methods. In our case, we'll use cell clusters to infer the cellular lineages. As you know, clustering analysis is a very difficult and somewhat frustrating question, because no matter what the data or clustering algorithm, we get clusters. So are these good clusters or not? The clustering results are often sensitive to the data and to the choice of algorithm. In practice, we try many clustering algorithms. The results are hard to validate, and there's a trade-off between cluster resolution and the replicability of the clusters across data sets. So you're probably asking, why are we proposing yet another clustering method? Well, we're not doing that. What we're doing actually is to provide a general and flexible framework to enable researchers to apply and compare a variety of different clustering algorithms and associated tuning parameters. And then based on all these different clustering, we generate a stable consensus clustering. This is our R-seq framework, which I'll describe next. I won't have time to describe Dune, but Dune is a cluster merging procedure to help us navigate the trade-off between cluster resolution and replicability across data set. The more clusters we have in general, the less replicable they have. So we need a way to find the sweet spot on that trade-off. So R-seq stands for resampling-based sequential ensemble clustering. It can be viewed as the unsupervised analog of ensemble methods in supervised learning, like bagging, boosting, and random forest. So ensemble clustering involves aggregating multiple clusterings obtained from different algorithms, or the application of a particular algorithm to resampled versions of the learning set. And it's a general way to improve stability. So here are the main steps in R-seq. We start from a base clustering algorithm, like partitioning around medoids or k-means and associated tuning parameters, and we get a clustering. Then we keep repeating that with different clustering algorithm. And then we compute a co-clustering matrix, and from this co-clustering matrix, we generate a consensus clustering. And then we refine the clustering by merging non-differential clusters, by testing for differential expression between sister nodes in this hierarchy of clusters. We also have a way of finding cluster signatures. So suppose you have 10 clusters. We want to find the genes that are differentially expressed between them. What comparisons do we do? We could look at one versus all, pairwise comparison, or a generic F-test. So that's not specific. Instead, what we recommend is using the hierarchy of cluster and then test contrast based on the hierarchy of clusters. And then we have various tools for visualizing the data. So this is when we apply the our seq framework to the P63 data set, we get clusters that correspond to a variety of states in the differentiation process of HBCs. So we have HBCs, of course, we have globose basal cell, microvillus cells, we have neuronal precursors, and we have immature and mature neurons and immature and mature sustentacular cells. So this is a pseudo-color image of the log counts for differentially expressed genes based on F statistic, a general F statistics that just looks for any difference between the clusters. So rows are genes here and columns are cells. So there's no real pattern going on. Instead, if we look at the same data, but now when we pick genes as differentially expressed based on contrast, based on the from the hierarchical tree, we get much more meaningful classification or clustering of the genes with patterns that help us differentiate between the different types of cells. So this brings me to lineage inference. So remember, we started from HBCs and wanted to study how these stem cells differentiate into other types of cell in the brain. So we've developed Slingshot for this purpose. It's a flexible and robust framework for inferring cell lineages and pseudotimes. Pseudotimes are sort of a way to measure time or distance along the transcriptome profiles of the cells. So Slingshot allows us to identify any number of novel lineages, and it's also a semi-supervised method in the sense that we have knowledge about certain parts of the process, like the terminal cell types. We can give that information to Slingshot. So there are two main steps here. The first step is to infer the global lineage structure. For this purpose, we work with cell clusters, and then we build a minimum spanning tree on these cell clusters. Once we have this coarse or global lineage structure, we map the cells and then we infer pseudotimes using simultaneous principal curves. So we applied Slingshot to our olfactory epithelium data set. We used the RC clustering, which I just showed you. And we supervised the end nodes. We told Slingshot that the end nodes were sustentacular cell, microvillus cells, and olfactory sensory neurons. And then Slingshot identified 3 lineages. One that was going from HBCs to sustentacular cell, one from HBCs to microvillus cells via globose basal cells, and then one that was going to olfactory sensory neurons. And these lineages have very different structures and very different patterns of gene expression. It turns out that in the first lineage, the sustentacular cell are produced via direct conversion of the HBCs. There's no cell division involved. By contrast, with the second and third lineages, there's these end states, that microvillus and neuronal cells, are produced via an intermediate proliferatory state. So here is a diagram of the results from Slingshot. So this is 3-dimensional principal components. The points on the graph are cells. They're colored by cluster. We build the minimum spanning tree, and then we have the simultaneous principal curves to identify the pseudotimes and map the cells to the lineages. So here are 3 lineages: sustentacular cell, microvillus cells, and olfactory sensory neurons. So downstream of trajectory inference, it's important to identify the genes that are associated with the lineages in order to illuminate the biological processes that underlie differentiation. So most approaches proposed to date assess differential expression between clusters. That's a discrete form of differential expression, which does not rely on these smooth patterns of expression that we've seen in the lineage inference. What we've tried to do in our work on trait-seq is to use generalized additive models with a negative binomial distribution to allow flexible inference of differential expression both within lineages as well as between lineages. And we can identify different types of differential expression patterns by testing null hypotheses that involve linear combinations of the negative binomial GAM coefficients. And we can also use the fitted values from these NB GAMs to generate clusters based on their expression patterns. So this is a standard generalized additive model. The mean is parameterized using cubic splines, and these are the coefficients that we focus on when we look for differential expression patterns. So it's probably clear to jump in this chart here that lists the different types of tests that we perform within TraitSeq. We have 2 tests for looking for differential expression within a lineage, and 3 tests for looking for differential expression between lineages. And here are 6 different case studies and the different types of D patterns that we would pick. So here there's no difference between the 2 lineages, but there's differential expression within the lineages. And we capture these different patterns again by looking for linear combinations of the GAM. So here are the results of TraitSeq on the OE dataset. So again, our 3-dimensional principal component analysis. We have the 3 lineages. The cells are colored by cluster. And this is a heat map of genes that were found to be differentially expressed along the neuronal lineages. And you can see 4 clear clusters of, sorry, 5, there's a small one, 5 clear clusters of genes here that correspond to different types of activity during the developmental process. We also identified 4 transcription factored by another type of test, the early DE tests that are involved in epithelial cell differentiation. And this is what's represented here in panel D. And finally, by looking at the first derivative of the GAM, we were also able to identify a transcription factor activity cascade, which is represented here in this figure. So I believe I'm out of time here. There's lots to say about the biology, and it's been absolutely wonderful to work with our collaborators in the lab. It's really motivated some really fun work and lots of methodological development to be done. So thank you very much for your time, and I look forward to the discussion.
Thank you for a great talk. So we have a couple questions here from the participants, and I also have questions of my own. So we have Malco Wu here, who says, hi, Professor Dudoit. Is it possible to cluster the same data set into different set of clusters? Each set represents an individual latent pattern with its own distinguished point of view. Thank you.
Yes, first, good morning, everyone. It's really a pleasure to participate in this workshop. I just wish we could see each other face to face, but yeah, this is great. So thanks for inviting me, and thanks for the question. So yes, I mean, the answer to the question from the participant is yes. In principle, we could produce different types of cluster that focus on different features of the data. So I think the main question here is really in collaboration with the biologists that designed this study to figure out what sort of patterns they're looking for, and then adapt the clustering procedure to zero in onto these latent patterns. So it all goes back to the question that you're asking and what you want to find from the data. So in principle, yes. Thanks for the good question.
So I guess as a follow-up to that question, I have a question, which is, you probably didn't have time. You only had half an hour, and you mentioned at the end that if you would have more time, you would talk about the biological implication of the research. I'm very curious. Those latent patterns that you will discover, what are the implications of that? Like, are there any application in health, or is it more understanding, just understanding how the body works in general, or what are the potential findings in real life of this work?
Yeah. Well, I'm really glad you're asking this question, because it's true, I only had half an hour to present this. So this study is part of a very ambitious plan that biologists have had for decades or centuries, which is to understand the brain broadly, right? But we adopt a divide and conquer approach to this. So our collaborator, John Nye, is a neurobiologist. He's a specialist of the olfactory system. So he's approaching this big, very general puzzle using this smaller system and examining in detail. So one of the applications of this study is to aging. So I didn't show this data set, but we also have similar expression data looking at aging processes and also genetic processes following injury. So this has down the road implications to understanding neurodegenerative diseases and eventually find therapeutics for ailments like Alzheimer's disease. So we're starting with sort of fundamental science problem, but that have very significant medical implication and medical benefits down the road. So we're focusing on a smaller system, a smaller model system to address these big questions.
Interesting, very interesting. So we have Isabelle Guyon here asking, do you have a way of testing the significance of cluster, or is this more human judgment?
Oh, that's a tough question. I think it's a mix. I think, as you know, a lot of clustering has to do a bit with an art. So there's a lot of human judgment, but eventually we want to also build processes for testing the significance of cluster. And that, you know, it's a harder question when we're in the unsupervised setting, but by having replication, there's ways to assess, you know, the clusters from a more statistical point of view. So the idea would be to do replications or resampling for that. Yeah,
I would assume that will allow to use some of that supervision in the clustering algorithms themselves, right? If
exactly. Yeah,
great. Colin Goodall is asking, you mentioned a number of R packages, many specialized to genomic data. Which among those packages, which package do you recommend for use with non-genomic data in R?
Oh yeah, so there's it's hard to navigate all the packages that are out there, that's for sure. So I'm mostly an R user. So for packages that are very generic, that don't focus on any specific types of data, I would go to the Comprehensive R Archive Network. So there's a range of packages for cluster analysis, for prediction, so you know, both unsupervised and supervised learning. I should say that also the packages that my group has developed, although we released them on this biological project, the packages can be used for more general data types. So for example, the clustering package, which we call Cluster Experiment, can be used on general data matrices. They're not linked to having expression data.
Great, thanks. Now we have Bibek Podel, who has two questions. First question, I'd love to know more about utilizing different modalities of data, for instance, RNA-seq and cellular expression. I was wondering if your group has been looking at this problem.
Different multimodal, I'm not quite sure I understand the question, the multimodal aspect.
Yeah, so maybe I can ask the next question from Vivek, and if Vivek wants to maybe precise what they mean by different modalities, we could follow up on that one. So let's go to the next question for now. Another question. I am working on this problem, and I find that the curse of dimensionality and the number of statistical tests grow up by a lot in single-cell data sets, especially if you do multimodal data sets. In your opinion, what is a good way to address this challenge using machine learning methods?
Well, that's a very broad question, right? That applies to many, many different fields. So, well, I'm going to answer with a very broad question. Because I was just reading about that recently, there's a fairly new field which is called post-selection inference that can help you broadly navigate this trade-off. So, you know, as opposed to looking in a completely agnostic manner and performing, say, hundreds or thousands of tests, you can do it in a data adaptive manner that sort of, you know, uses the data in subpart to zero in on the comparisons you want to make on the prediction you want to make, and accounts for that in terms of how it the procedure assesses false positive error rates or prediction error. So there are fields like that. So of course the multiples nesting literature tries to address that, but more recently post-selection inference. And I could send, there's a group of young faculty in my department that has been working on that, and it seems like a really promising field. And they have a weekly seminar to address that. So a lot of cutting edge development going on in that field.
Thanks again for a wonderful talk. That's all the questions we have. So we will go to a break and restart again in 15 minutes. So thanks again, Sandrine. And
well, thank you for having me over. It's a pleasure.
Yeah.